Hi, and thanks for checking in on Beneath the Surface Day 4. So some of this video you might have seen if you watched the Beneath the Surface Day 3 video. I have some overlap here. So if you get to parts that you've already heard me talk about, particularly limiting factors and rewilding, go ahead and fast forward through or skip through some of that video so that you don't feel like you're watching the same thing twice. All right. So today we used pencil and computer. As a warm-up, we remember that two abiotic factors that kept our classroom bear population from thriving when we played our bear game the other day. Abiotic, again, is going to mean not alive, never alive. So we had polluted water. There were chemicals in the water from our chemical plant. And also fire. Those are abiotic factors that kept our bear population from thriving. Two biotic factors that hindered the classroom bear population. Those are two things that are alive. We talked about several things that were alive. So bears competing for space with each other. Those are biotic factors. The fact that there was less woods, basically less den material available, that was a biotic factor. Humans hunting bears, that is a biotic factor. We had our juvenile bears being inflicted by some disease, and so disease would be a bacteria or something like that, and so they were, that was a biotic factor. And then, of course, if there was not enough food, that would have been biotic. All right, so those were some of the things that we are now going to say are limiting factors because they limit the number of bears that can survive. But I want you to keep some things in mind from previous science days. So if we look at question 3, it says, which domain contains organisms that are prokaryotic and can live in extreme environments? So our big hint here is those extreme environments, and we mean really salty areas or areas that have a lot of sulfur or they're extremely high temperatures. Those, if you remember, are going to be from the domain Archaea. All right, so at this point, if you watched the Beneath the Surface Day 3 material, you will have seen some of this. So you're welcome to fast forward through. Just make sure you don't miss anything new. We were able to finish limiting factor in 5th period, but not in all of the class periods, so I'm just doing it again. All right, so rabbits in the field. Female cottontail rabbits, this is the Sylvolagus floridanus. If you look right here, that's the scientific name. Remember, with a scientific name, it tells us genus and species, and we've typed it in italics so that it's slanted looking. We have a capital letter on our genus, but a lowercase on the species. Okay, so female cottontail rabbits are especially fertile. They are able to give birth to 7 litters per year. And this might suggest areas would be overrun with cottontail rabbits, but that isn't actually the case. So we'll try to talk about some things that might be limiting the growth or overgrowth of our cottontail rabbits. All right, so you can watch this video and be thinking about answering what a limiting factor is and what 2 examples of limiting factors are from our video. But after the video, we'll actually pause and put some official definitions down too. So let me see if I can fix my audio. Hold on. I'm going to pause.
Welcome to Moo Math and Science. In this video, I'd like to talk about limiting factors. Any abiotic factor or biotic factor that restricts the number or reproduction of organisms in an ecosystem is called a limiting factor. Take this ecosystem found in the fishbowl. The limiting factors could be the size of the bowl, the amount of water, or the temperature, and any of these can limit the number of fish that can survive in the fishbowl. Limiting factors can be dependent limiting factors and independent limiting factors. Dependent limiting factors depend on the number of organisms. For example, the amount of food available for each organism depends on the number of organisms in this ecosystem. And independent limiting factors does not depend on the number of organisms. For example, the amount of rainfall does not depend on the number of organisms. You also have density-dependent limiting factors, and it is a limiting factor that depends on population size. For example, these could include competition, predation, parasitism, and disease. Let's look at a list of abiotic limiting factors. This could include sunlight, climate, temperature, water, space, soil chemistry, fire, and natural disasters. And a list of biotic limiting factors include the number of plants, number of animals, amount of competition, number of decomposers, number of parasites, and disease-causing agents. So there we go, limiting factors, biotic and abiotic factors that limit the size, number, and reproduction of organisms in an ecosystem. Thanks for watching, and Moo Math uploads a new math and science video every day.
Okay, so what is one abiotic limiting factor for the fish in this fish bowl? Well, we might say space. We might say water. The amount of, let's see. Mostly I think I'd say water and space for this one. What could a biotic limiting factor be for organisms in the fish bowl? That could be competition. So if I have more fish in there, especially if one fish is going to eat another, those are biotic limiting factors. Food, for sure. The plants in there, if they are being eaten for food, those would be biotic limiting factors. But if we need oxygen in the water so that the fish can breathe, because they actually do breathe oxygen through their gills, then we might say that sunlight is an abiotic limiting factor, and also oxygen would be an abiotic limiting factor. All right, so don't write these two things down. You can just listen to two different definitions of limiting factor. So limiting factors are conditions of the environment that limit the growth of a species. So that could be too much sun or not enough sun. It could be temperature. It could be several different things. So if we look at the second definition down here, another definition for limiting factors is biotic and abiotic factors that prevent the continuous growth of a population. Something that is going to limit the number of organisms that can continue to grow. All right, so here's your official limiting factors definitions and examples. And if you were in class with your bare tally sheet, this information is right on the back side of that page. Third period, I actually gave you just a new copy of just this page. So limiting factor is, oh my goodness, look at that typo, I'm so sorry. A limiting factor is something that controls the growth of a population in an ecosystem. Examples of abiotic limiting factors, that means not living things that are limiting, could be oxygen, space, sunlight, water. Could be pH, which is essentially the acidity or basicity. So water can, or liquids can be basic or acidic. And we like them to be pretty close to neutral, just like you wouldn't want to put your hand in any kind of a heavy acid. You wouldn't want to put your hand in any liquid that is considered a severe base either, because that's just going to damage your skin. So pH is another abiotic limiting factor that's not written there. And it has to do with per hydrogens, just to know what the P and the H are for. All right, so looking down here, examples of biotic limiting factors, those could be predators. And predators could be bigger animals. So if we're not talking about bears, if we're talking about bunnies, we might be thinking that foxes and I imagine eagles and hawks even, probably big birds eat bunnies and things. Those would be predators. Hunting, that's going to be humans hunting animals, but that definitely would be a biotic limiting factor. And then disease or illness of some sort. All right, so again, one more time. What is a limiting factor? It's anything biotic or abiotic which keeps a population from growing out of control. All right, there are other things that affect our ecosystems. So we know that if we were to look at the Earth 200 years from now, it's not going to look exactly the same as this. And that's partly because we know that the continental plates are shifting with continental drift. We know that our water levels are rising over time because of climate change. So we know that the Earth would look different if we were to have a fancy new picture 200 years in the future. So we're going to look at two pictures of the same basic location and think about what we're noticing. So you can see that there's a lot of dirt here on the right side where there used to be structures and roads and things on the left. This is our before picture. This is our after picture. So I notice that there are houses missing. There's a lot of dirt here. There aren't really the trees and bushes that used to be here. And then if I want to think about what might have caused this, could be any number of things. I don't have enough information, but I can imagine that maybe there was a mudslide, maybe there was a dam that broke uphill from this area, or maybe there was a little bit of a natural disaster like a tornado or something. There are lots of things that might have happened here, and so I could speculate, but I'm using my inferences, my background knowledge to imagine what might have happened. But if I want to think about how the environment would be different from one picture to another, well, there would probably be more little critters living in the before section. Certainly there are more plants in this before section here. So we have a higher biodiversity before compared to after. And remember, higher biodiversity is better. It's a healthier ecosystem when it has a higher biodiversity. Here we've lost a lot of bushes and trees, and so we probably don't have quite as many groundhogs and rabbits and little things running around and hiding in there. And we certainly don't have as many plants growing. All right, so here's a different before and after scenario. They are probably slightly different seasons, but it's the same location. And so this was in 1999 on the left, and this is 2015 on the right. So what we notice is that there's a whole lot more plant life, more types of plants in the after picture. You can see different textures. It's not just that it's a different season. It really does look very different. And so I wanted to talk about why that might have happened in this ecosystem. If we have trees and plants that have changed, we can imagine that there are probably animals that have either been able to grow more profusely, or more animals have come and made this their habitat because they have more places to hide and more plants to eat. So humans do this on occasion to ecosystems that are lacking in biodiversity, and when they improve a situation, it's called rewilding. This term rewilding is just the reintroduction of species of plants and animals to an area, generally to try to bring it back to a higher biodiversity area. So if you think about something like Mount St. Helens, that's another place where there have been rewilding efforts. After Mount St. Helens erupted in the 80s, a lot of plants, of course, died and animals too. And so there have been rewilding efforts there to try to improve the plant cover and therefore the species of animals that exist there too. So rewilding is just a term that means reintroducing species of plants and animals to an area to try to improve it. All right. So just remember, our limiting factors are things that prevent continuous growth of a population. So let's think, are they always a bad thing? Why or why not? Well, they're not always a bad thing, because if you think, sometimes we have so many deer in this Northern Virginia area that they are competing for food and they all start to get a little bit sickly. And so they are competing for food, they're getting sick, they are getting hit by cars because there are so many and they're just kind of running across the street and things. So we have programs where we try to help eliminate some of those deer. And by doing so, we actually make the ones that are still surviving healthier because they have more food available to them, and they don't have such overcrowding. And when we think back to our bunnies, there's natural limiting factors of other things like foxes that are eating those bunnies. And they naturally keep that population down, because if we had too many bunnies, those bunnies would also not be a healthy population if they were overcrowding. All right, so in class we played a Kahoot, and if I can figure out how to make that a play individually, I'll add that to Schoology, but I'm not 100% sure I can. It was just some review of our terms like biotic and abiotic and ecosystem, limiting factor, things like that. All right, so if you'd like to try to unscramble these science terms, you can pause the video and then come back. All right, so hopefully you paused. This turned into bacteria. This was domain, kingdom. This was safety, as in lab safety, abiotic, and this was ecosystem. So just for a little brain break, we tried to rearrange those words and come up with our science terms. All right, so if you're able to, you can do that Kahoot very quickly as a review if I'm able to get it into Schoology. But otherwise, you can go ahead and complete the exit ticket for day 4. You may use your notes and you may try twice. So just remember, pH is an abiotic factor that has to do with the acidity of a liquid. All right. Then, after we did that exit ticket, I asked everyone to complete a gizmos. So this is our first time trying to do a gizmos in class. I handed out a worksheet, and that worksheet is available if you look in Schoology at the Coral Reef Assignment link. You'll find the PDF, the worksheet that we used. When we use gizmos, I'm going to skip over this for a second. You're going to go to Schoology, click on the waffle icon, and then you're going to choose Clever. And I swear I have this. There you go. I knew I had this in here. This is Miss Presigil's, and mine works exactly the same way, so yours just looks a little different because it's got my icons. So you're going to go into your Schoology, and you're going to click the waffle, choose Clever, log in with Google, and then you should see this Gizmos icon, and you're going to click on that. You might have to go into our class, but most of the time people were able to click right on that Gizmos icon. So when you click on the Gizmos icon, then you're going to click on Coral Reefs, Abiotic Factors. It should be the only one that's there, and click on Launch. Okay, I'm going to back up. Sorry to make you dizzy. I'm going to back up for a sec. So if we're thinking about an abiotic factor in a coral reef that a biotic factor would depend on, we might be thinking about the temperature of the water or the salinity. That's how much salt is in the water. And if we are thinking about what a limiting factor for the fish population could be in a coral reef, if you have less coral, the fish have fewer places to hide. If you have more predators, that could limit the fish populations because maybe the smaller fish get eaten. And again, the fish population could be limited by temperature or salinity as well. So, all right, we're going to see what happens to organisms in a coral reef when there are abiotic factor changes. So if you would please make sure that you are in your gizmos, and I just want you to notice we've got coral reef tab, we've got a data tab, we've got a conditions tab, and we've got a summary tab. Also notice where it says ocean conditions here, there's an arrow. You can click on that arrow and it's a pull down menu that gives you some other options. You'll be using those toward the end of your practice with this. When I'm looking at my image of a coral reef, when you see it, you'll see these fish moving and the turtle moving. You can click on any one of these organisms, and if you click on it, you'll learn more about it. Okay? Right now, if there's something to read, I would say generally read it. This says corals are affected by storms, water temperature, and pH, and high temperatures may cause corals to bleach or lose their symbiotic algae. So it's just like bleaching your shirt. It just changes the color. All right, notice that we have a button to return to original settings. So if we have changed the pH to make it higher or lower, if we change the ocean temperature, if we change the storm severity, we can change all of those things and then we can advance years. And normally I would go at least 10 years. You just click on that 10 times and then you can see what changes happened. You can look and see, is there more algae or less algae? These lighter greens are algae. Is there more of this staghorn coral or less? And then there's a star boulder coral, I think that is. So you can get more information after you advance 10 years. And again, you can click on things and always read what is given to you because that's what you want to find out. When you're done and you've messed around and changed some things, you can always click to return to original settings and you can restart and you'll be back at year 0. Okay, so once you're playing around, it'll probably be easier for you to see some of those things. You've already done these steps. You're in Clever, you're in Gizmos, you have launched abiotic factors. So again, we are only looking right now at storm severity and ocean temperature and ocean pH. So we are looking at abiotic features, right? Okay, so just be aware of that. You can select any organism, click on it, and you'll learn more about it. So let's see what happens with that. These are my 2 corals. So if I click on one, I'm going to get information. And if I click on the other, I'll get information. I want you to think about which one right now, when we click on the 2, we're going to get information, which one might be able to survive strong storms better. So let's see. So if I click on the staghorn coral, it says it's the fastest growing and most delicate Caribbean coral. It obtains energy from photosynthetic symbiotic algae. So just realize algae are plants, algae are photosynthesizing, and they are working symbiotically with the coral to share energy. These particular staghorn corals are vulnerable to something called white band disease, and right now they are covering about 21% of the reef floor. Okay, staghorn coral, fastest growing, most delicate. The other guy, boulder star coral, is a robust coral species. It grows slowly. It also gets energy from photosynthetic symbiotic algae, and it's vulnerable to something called black band disease. It's currently covering about 28% of that reef floor. So remember, we had to predict which one of these 2 corals would be stronger in storms. I'm going to say it's the boulder star coral because this one's robust. The other one was delicate. All right, so you could click on conditions up here if you're not already on that tab. We're going to up our storm severity and we're going to advance our year and then see what happens after we made a prediction. Then you can always return back to original settings and restart. Okay, so right now at year 0, my coral reef is healthy with no bleaching or disease. The fish diversity is high and the coral cover is high. I can see all kinds of numbers and data right up here. Storm severity is at 20%. And my ocean temperature is 27. The pH is 8.1. You can see all this information, right? Coral cover is 49%. And the total fish species is 442. So we're going to click on summary. If you hadn't seen that, you'd click on summary and you can see what is the total coral cover. It's 49%. All right. Total number of fish species is 442. So now I want you to click on conditions, and we're going to up that storm severity to 50%. you can either click in here right where your original number was and type in 50, or you can drag this bar. We're going to advance 10 years. Click, 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 click 10 times. Okay, and so this is just a screenshot. You can't usually see them side by side, but just looking, you can see they're close, so I'm going to need to read the materials that are given to me. I can see those might be sponges or something. I'm not sure what those are because I am not interactive right this minute, but it looks like I don't have as much coral covering things, right? And so you can look at different species and things too. All right, so definitely when you're clicking around, make sure you look at the words that you are given because that's going to tell you some information about your new ecosystem. All right, remember you can always restart and you can always change your settings. Okay, so in addition to a summary, you can also go to this data. So this is really cool because it's going to give you graphs that show you how much things are growing or how much they are declining. You can select every one of these organisms. And so on your worksheet, it's going to ask you to do that. So if you're here and you've chosen some organisms to consider, you're going to advance the years. Make sure your first column is checked for sure. And you're going to advance your years 10 years again. And you're going to see changes here. Notice that I've got the percentage of the normal population compared to the year. Okay, and staghorn coral is written in red, so you're going to look for a red line for that coral. The boulder star coral is black, and so you're looking for a black line. Sponges have a yellow line, and algae has a green line. So click advanced year 10 times, and look and see what you notice. Okay. Hopefully you see that this red line has gone down, and so you look and say, what's red? Well, staghorn coral is red. So over 10 years, our population of staghorn coral has decreased a lot, right? I notice this green line coming straight across. That's the algae. It really hasn't changed at all. The sponges really haven't changed at all. And my boulder star coral has only gone down a little bit. So just realize you can click on coral reef, you can click on data, you can click on conditions, you can click on summary, you can restart and reset, and you can advance your years, and that's going to give you tons of information. Okay, summary, conditions, data, coral reef. All right, so remember we talked about the coral cover earlier. It was higher than it is now, and our fish species was higher than it is now. So you can write down actual amounts on your paper. All right, please now return to original settings and then restart. And you're going to use your worksheet to go ahead and explore how changing the ocean temperature affects the reef. Okay, just follow these directions as you go through, and make sure your name is on your paper. I forgot to give you a place to put your name, but follow the directions. They are relatively clear, and I say good luck, and this will be due next class. Okay. We did not talk about our Daphnia last class, so we will talk about Daphnia next class. And you are completing your worksheet. And we did have a brief moment where we remembered the difference between observations, and inferences. So the boy is in the water, that's an observation. That is a thing that exists. The tree branch is broken, that is true. That is an observation. The boy fell off the branch into the pond. Maybe. That might be what happened. So that's an inference. That depends. We don't know, right? Because also maybe the goat pushed the boy into the pond. He's looking like maybe he did. Or maybe the boy was hopping around in the rocks and fell into the pond. So these are three different inferences based on the same observations. All right. So you are completing your coral reef exploration. And just think about things that happened. Did anything else surprise you as that temperature increased? You can also always go back and advance lots more years if you want to see even bigger changes. Okay, so if you messed around with conditions like logging or land use, you might have been able to decrease your algae population, which is sometimes good, but that was probably something that only some of the class periods had time for. In your part 2, you do need to pick a land use or a fishing or a disease or invasive species. You're going to choose one of these 3 things in conditions. What did you change? I want you to tell me whether you raised or lowered something, one of these 3 things. Tell me what the new number is. And then describe one thing that happened. Remember, you can use the summary tab and the data tab. All right, so good luck with that, and email if you have questions. And thank you again for watching.